My lesson for today will be the second part of cellular respiration, and it will be about the plants, especially with the topic of photosynthesis. So, for the Earth's earliest life forms, it is believed that the Earth's earliest life forms obtained their raw materials and energy from simple molecules that are dissolved in aqueous environment. And these molecules had to have formed abiotically, or there are no presence of living organisms in chemical reactions, which occurred in the primeval or primitive seas. Thus, their survival were thought to be because of the presence of nutrients taken from the environment, like what you call your heterotrophs. So again, these types of organisms do depend on external source of organic compounds for them to be able to thrive. So the number of heterotrophic organisms living in the primitive Earth must have severely restricted because the spontaneous production of the organic molecules that occur very slowly. And because of this, the evolution of life on Earth received a tremendous boost with the appearance of organisms that employed a new metabolic strategy wherein they could manufacture their own organic nutrients from the simplest type of organic molecules like your carbon dioxide and hydrogen sulfide, which are called the autotrophs. So again, when it comes to the heterotrophs, they are very dependent on the external resources that they do have, while the autotrophs do have the capability of, producing their own food. So this slide would show you some examples that we have for the autotrophs and the heterotrophs. So again, for the autotrophs, usually they are the producers. They have the capability to, produce their own food via photosynthesis or chemosynthesis. So we have examples of plants, some bacteria and algae. And then for the heterotrophs, usually they are the consumers that eat other organisms to get proteins and energy. For example, animals, most of the bacteria, and your fungi. So in addition to the autotrophs, they were actually considered to require much greater amount of energy to manufacture complex molecules from carbon dioxide. So because of this, they evolved in such a way that their types were identified based on their source of energy. So the first one is the chemoautotrophs. So they utilize the chemical energy stored in organic molecules such as ammonia, hydrogen sulfide, or nitrites to convert carbon dioxide into organic compounds, which are usually present in your prokaryotes. And then the second one, we have the photoautotrophs. They actually use the radiant energy of the sun to achieve the same result. And these photoautotrophs are also responsible for capturing the energy that fuels the activities of most organisms on Earth. And it includes plants and eukaryotic algae, various flagellated protists, and members of several groups of the prokaryotes. So basically, these photoautotrophs, they are organisms that do carry out the process of photosynthesis. With photosynthesis, it is in which the energy from the sunlight is transformed into chemical energy that is stored in carbohydrates and other organic molecules or metabolites. So during photosynthesis, relative low energy electrons are removed from a donor compound and converted into high energy electrons using the energy absorbed from light. With this, high energy electrons are then employed in the synthesis of reduced biological molecules such as your starch and oils. With your early photoautotrophs that may have dominated the Earth for 2 billion years, they actually used your hydrogen sulfide as their source of electrons for photosynthesis with the reaction carbon dioxide plus 2 moles of hydrogen sulfide synthesized with light, resulting to a unit of carbohydrate plus 2 moles of sulfur. Okay. So, however, because hydrogen sulfide is not usually abundant or widespread, the organisms that depend on it as an electron source are already restricted to habitats that are rich in sulfur, like sulfur springs and deep sea vents. So in here, you will see the green sulfur bacteria, which is one of the numerous bacteria living today that carry out the photosynthesis using hydrogen sulfide. And then a new type of photosynthetic prokaryote appeared on Earth that was able to utilize water. Because actually water is more abundant resource that we have on Earth, and basically this is also a resource wherein it can be, or it can give a huge amount of electrons. So with this one, this prokaryote that we are actually talking about is what we call your cyanobacteria, that appeared in much more diverse habitat and produce more waste products with consequences for all the forms. So with the cyanobacteria evolving, there are also other effects that were actually developed, or that actually developed because of their existence. So waste product was molecular oxygen, which is formed in the overall reaction that we have here on your screen right now, which is the carbon dioxide plus water synthesized with light results to carbohydrates plus an oxygen atom. So in here, if an organism is going to carry out oxygen, or technically being called as oxygen releasing photosynthesis, it has to generate a very strong oxidizing agent as part of its photosynthetic metabolism in order to pull tightly held electrons from water. At some point in time, one of these ancient oxygen-producing cyanobacteria took up residence inside a mitochondria containing non-photosynthetic primitive eukaryotic cell. So, over a long period of evolution, the symbiotic cyanobacterium was transformed from a separate organism living within a host cell into a cytoplasmic organelle, the chloroplast. So, as the chloroplast evolved, most of the genes that were originally present in the symbiotic cyanobacterium were either lost or transferred into the cell nucleus. As a result, the polypeptides found within a modern-day chloroplast are encoded by both the nuclear and chloroplast genomes. So, for the use of the chloroplast, it was identified in 1881 by Engelmann as the site of photosynthesis. So, he illuminated cells of the green alga Spirogyra and found that actively moving bacteria would collect outside the cell near the site of the large ribbon-like chloroplast. And then the bacteria were utilizing the minute quantities of oxygen released during photosynthesis in the chloroplast for their aerobic respiration. So the image that you see right now on your screen is actually what you call your filamentous alga Spirogyra. So this is actually how it looked like. So for the functional organization of your chloroplast, chloroplasts are actually located predominantly in the mesophyll cells of the leaves around the cell's central vacuole. So here is the mesophyll, and then you will be able to see here your palisade cells. And then having or focusing on these cells inside of it, you actually see there your chloroplasts. Okay, so the structure of the leaf and the arrangement of the chloroplast around the central vacuole of the mesophyll is again shown in the figure on your right. So chloroplasts of higher plants are generally lens-shaped. They are approximately around 2 to 4 micrometers wide and 5 to 10 micrometers long, and typically numbering 20 to 40 per cell. So they arise by fission from pre-existing chloroplasts, or either non-pigmented precursors, which are called your proplastids. The outer covering of the chloroplast consists of an envelope composed of two membranes separated by a narrow space. In here, figure 6.3a actually shows the TEM image of the chloroplast wherein the internal membrane is arranged in stacks, or what you call the grana of disc-like thylakoids, and the shape of your chloroplast, which is lens shape. Okay, so inside you will see here the stroma and then the thylakoids. Okay. In figure 6.3b, this is a schematic diagram of a chloroplast showing the double membrane and the thylakoid membranes. So in here you will see that you have an outer envelope membrane and an inner envelope membrane, and of course the stroma and your thylakoids. So, like the outer membrane of a mitochondrion, the outer membrane of a chloroplast envelope contains several different porins. So, these porins do have relatively large channels, but they exhibit selectivity towards various solutes and thus may not be as freely permeable to key metabolites. So even if there are so many porins on this membrane, again, they can be selective in some ways. Okay? Especially when it comes to the solutes that may, go through the membrane. So the inner membrane of the envelope is highly impermeable, and substances moving through this membrane do so only with the aid of a variety of transporters. So if you compare it also with your plasma membrane, they do also have their transporters that would actually help let other molecules go in and out of this plant cells. Okay, so much of the photosynthetic machinery of the chloroplast, including light-absorbing pigments, a complex chain of electron carriers, and an ATP synthesizing apparatus, is part of an internal membrane system that is physically separate from the double-layered envelope. So next, let's move on to the stroma of the chloroplast. So the stroma of the chloroplast contains small double-stranded circular DNA and prokaryotic-like ribosomes. So basically, this is where they store their genetic information for plants, which is actually similar to the matrix of your mitochondria. So the DNA of the chloroplast is actually a relic of the genome of an ancient bacterial endosymbiont. Depending on the organism, chloroplast DNA contains between about 60 and 200 genes involved in either gene expression or photosynthesis. And then next, we have your thylakoid membrane, which have a high protein content and are unusual in having relatively little phospholipid. So basically, they have high percentage of galactose-containing glycolipids. And both fatty acids of these lipids contain several double bonds, which makes the lipid bilayer of thylakoid membranes highly fluid that facilitates lateral diffusion of protein complexes through the membrane during photosynthesis. So next, let's go now to the overview of photosynthetic metabolism. So in here, photosynthesis has similarities to aerobic respiration, but it is actually the process in reverse that requires energy. So C.B. Vaniel in 1931, a Stanford grad student, proposed the understanding of chemical reactions of photosynthesis. But before that, people before him believed that the energy of light was used to split carbon dioxide, in return releasing molecular oxygen and transferring the carbon atom to a molecule of water to form a unit of carbohydrates. But then, in the time of Samuel Ruben and Martin Kamen, the role of water in molecular oxygen has been clearly established. So they carried out experiments on suspensions of green algae using a specially labeled isotope of oxygen, which is isotope oxygen number 18, as a replacement for the common isotope oxygen-16. So both experiments proved actually that oxygen is being split contrary to the popular belief, which is actually they believe that it was primarily carbon dioxide, but the notions um, were incorrect. So they were able to prove that it was oxygen being split, and Van Niel's proposal was replaced, where respiration in mitochondria reduces oxygen to water, and photosynthesis in chloroplast oxidizes water to oxygen. So with the initial photosynthetic reaction, it was known to be carbon dioxide plus water with light would actually produce um, carbohydrate atom and oxygen. Okay? So, the events of photosynthesis can be divided into two series of reactions. So, the first one would be the light-dependent or the light reaction. So, this is again the first stage of photosynthesis, wherein energy from sunlight is absorbed and stored as chemical energy into two key biological molecules we know as ATP and NADPH. So ATP is the cell's primary source of chemical energy, and NADPH is its primary source of reducing power. And then the next one is the light independent or the dark reactions, wherein carbohydrates are made from carbon dioxide using the energy stored in ATP and NADPH molecules produced by the light dependent reactions. In actual fact, these dark reactions take place much more rapidly when the light dependent reactions are also occurring. Okay, so basically both light dependent and independent dark reactions do actually work together hand in hand. Okay, so let's tackle first what are or how the light dependent reaction happens. So basically, when you say light dependent reactions, these are reactions wherein there's the absorption of light. So light energy depends on the wavelength. This is usually depicted with the lambda sign, and they travel in packets you call the quanta, cold photons. And the first step in any photochemical process is the absorption of light. Okay, so again, we have the wavelength depicted by lambda, the packets you call quanta, and then of course the presence of the photons. Okay, so when there are photons, it just means that there is light absorption. Okay, so a molecule is said to have shifted from the ground state to the excited state if an electron becomes sufficiently energetic to be pushed from an inner to an outer orbital when a photon is absorbed by a molecule. So again, what happens if your electrons get excited? So let's have here an example, which is our chlorophyll, which is the most important light absorbing photosynthetic pigment. So if the excited electron drops back to a lower orbital, the energy it absorbed must be released in the form of light, or fluorescence, or phosphorescence, or heat. With this one, it will give the chlorophyll, or the chlorophyll will have to return to the ground state, and photon is basically not utilized. Okay, so for intact chloroplast, energetic electrons are passed to electron acceptors before they drop back to lower energy orbitals. So next, let's go to the pigments. Your pigments are actually the colored molecules that absorb light of particular wavelengths within the visible spectrum. So pigments contain chromophore, a chemical group that absorbs certain wavelengths in the visible spectrum. And plants contain lots of chlorophyll, which absorbs red and blue, leaving the intermediate green wavelengths to be reflected to our eyes. So this actually makes, the plants specifically green in color in our vision. Okay? So the chlorophyll structure contains or consists of two parts. So the first one would be your porphyrin ring. So this is the porphyrin ring that you see here at the top or in the image on your screen right now. So porphyrin ring absorbs the light. So basically the function is for light absorption. And then you have your hydrophobic phytol chain that's keeping the chlorophyll embedded in the photosynthetic membrane. So this phytol chain is actually derived from the repeating isoprenoid units. To continue, so let's have or discuss about the porphyrin ring. So the porphyrin ring is composed of magnesium atom, unlike the one that you see in your red iron containing porphyrins in the blood or the heme groups of myoglobin and hemoglobin. So this is different from that type of porphyrin. So the alternating single and double bonds along the edge of the porphyrin ring, the localized electrons which form a cloud around the ring. So from what you have seen a while ago from the image, there is like a cloud around, cloud formation around the ring. And then systems of this type are said to be conjugated and are strong absorbers of visible light. So an absorption spectrum is a plot of the intensity of the light absorbed relative to its wavelength, and the range of wavelengths absorbed by photosynthetic pigments situated within the thylakoids is further increased because the pigments are non-covalently associated with variety of different polypeptides. So there are several classes of chlorophyll differing from one another in the side groups attached to the porphyrin ring, and this occur among photosynthetic organisms. So chlorophylls are the primary, they are again the primary light absorbing photosynthetic pigments, but we have here several different types of chlorophylls. So the first one would be chlorophyll A. This is found in all oxygen-producing photosynthetic organisms. So they are missing from sulfur bacteria. We also have chlorophyll B. These are found in all higher plants and green algae. And then chlorophyll C, they are usually found in brown algae, diatoms, and certain protozoa. And then we have the bacteriochlorophyll, which are only found in green and purple bacteria that do photosynthesize without oxygen formation. So those are the different types of chlorophyll. So we also have what you call your accessory pigments. So these accessory pigments are found in terrestrial plants, which also contain orange and red accessory pigments you call your carotenoids, which includes your beta-carotene. So this specific type of accessory pigment produces an orange and red colors in carrots, oranges, and some autumn leaves. And carotenoids can absorb primarily blue and green light, reflecting orange and yellow and red light, and they contain linear system of conjugated double bonds. So carotenoids have actually multiple functions. The first is they act as secondary light collectors during photosynthesis, And the second one is that they draw excess energy away from excited chlorophylls and dissipate it as heat. If not, energy may be transferred to oxygen, making an ultra-reactive singlet oxygen that destroys biomolecules and causes cell death. So because of the light falling on a leaf is composed of a variety of wavelengths, the presence of pigments with varying absorption properties ensures that a greater percentage of incoming photons will stimulate photosynthesis. So here you will see in figure 6.7 the absorption spectrum for several photosynthetic pigments of higher plants. So in here you will see the absorbance rate and of course the wavelength, which is measured in nanometers. So the background shows the colors that we perceive for the wavelengths of the visible spectrum. And then, of course, the chlorophylls absorb most strongly in the violet, blue, and red regions of the spectrum, while the carotenoids also absorb into the green region. And then red algae and cyanobacteria contain additional pigments like your phycobilins that absorb in the middle bands of the spectrum. On the other hand, we have figure 6.8 showing you the action spectrum of the photosynthesis. So the red line indicates the relative efficiency which light of various wavelengths is able to promote photosynthesis in the leaves of a plant. And we have there also the black lines which indicate the absorption spectra of each of the major photosynthetic pigments. The green line shows the combined absorption spectrum of all pigments. Okay? So next, let's go to the photosynthetic reactions and reaction centers. So in 1932, Emerson and William Arnold of the California Institute of Technology carried out an experiment suggesting that not all of the chlorophyll molecules in chloroplast were directly involved in conversion of light energy into chemical energy. Okay, so basically in this one, they are just saying that not all of the chlorophylls or the chloroplast in plants are involved in the production of in the conversion of light energy to chemical energy. So how were they able to do it? So their experiment actually used suspensions of the green alga chlorella and flashing lights of extremely short durations around 10 microseconds at saturating intensity, and they determined that the minimum amount of light needed to produce maximal oxygen production during photosynthesis. So based on the number of chlorophyll molecules present in the preparation, they calculated that one molecule of oxygen was being released during a brief flash of light for every 2,500 molecules of chlorophyll present. So basically, there can only be, or in in um, in easier ways to understand is that if there are 2,500 molecules of chlorophyll, in that 2,500, it can only produce one molecule of oxygen. Okay, so this already gives you the idea that what they initially thought of is actually correct. That not all of the chloroplasts are actually involved in the conversion of light into chemical energy. Because pertaining to the amount of chlorophyll present, they can only produce one molecule in a brief flash of light. Okay, so eventually, um, they have concluded that 300 or so chlorophylls act together as one photosynthetic unit in which only one member of the group, or the reaction-centric chlorophyll, actually transfers electrons to an electron acceptor. Okay? So even though the bulk of the pigment molecules do not participate directly in the conversion of light energy into chemical energy, they are still responsible for the light absorption. So these pigment molecules form a light harvesting antenna that absorbs photons of varying wavelength and transfers that energy called excitation energy very rapidly to the pigment molecule at the reaction center. So again, you see here that there are antennas that are being produced in this transfer of excitation energy. So in the middle, you will see the reaction centers, and of course, you will see the introduction of photons or the absorption of light energy. Okay, so chlorophylls of antenna are kept in close proximity with less than 1 nanometers apart and proper orientation by non-covalent linkage to integral membrane polypeptides. So this is very important that these antenna are very, very close with each other. Okay, so a rule that operates among antenna pigments is that energy can only be transferred to an equal or less energy requiring molecule. So this is why you see patterns on the image, right? So basically, again, the passing of the energy can only be sent to those who really require it. Okay? So as energy passes through the photosynthetic unit, it is repeatedly transferred to a pigment molecule that absorbs a longer wavelength. So energy is lost and nature of future transfers become restricted. So finally, energy gets to reaction center chlorophyll, wherein there is an absorption longer wavelength, lower energy light than any of its neighbors, and passage from the absorption site to reaction center takes around 200 by 10 to the 12th power second. So the reaction center serves as an energy sink in the photosynthetic unit, and once the energy is received by the reaction center, the electron excited by light absorption can be transferred to its acceptor. Okay. So next we have the oxygen evolution. We will be discussing further the coordination of the action of two different photosynthetic systems. Okay. So the evolution of organisms capable of using water as an electron source was accompanied by major changes in the photosynthetic machinery. So the reason for changes is revealed by considering oxygenic photosynthesis energenics, or those that are capable of releasing oxygen. And basically, 1 mole of photon around 680 nanometers wavelength, equivalent to red light, is equivalent to a 1.8 volt change in redox potential. So the photosynthetic light absorption occurs in large pigment protein complexes you call photosystems, and there are actually two photosystems that is present or that's available. Okay, so the first one is the photosystem 1 or the PS1, and the second one is the photosystem 2 or the PS2. So both actually catalyze the two light absorbing step. So PS1 and PS2 mediate distinctly different photochemical reactions but exhibit marked similarities in protein composition and overall architecture. So in here, P60 or pigment 680 is PS2 reaction center. Okay? So again, it's very important for you to remember that P60 is the reaction center for the PS2 photosystem. Okay? So basically, this P680 absorbs light mostly strongly at around 680 nanometers. Okay? And P700 or pigment 700 is actually for the PS1 reaction center, which absorbs light most strongly at 700 nanometers. Okay? So, in oxygenic photosynthesis, where two photosystems act in series, electrons flow along chains of carriers towards positive reaction centers, and the chain actually consists of three legs, and collectively they are known as the Z-scheme. Okay, so the three legs that are being considered here is the first one is from water to the photosystem 2. The second one would be from photosystem 2 to photosystem 1, and then lastly from photosystem 1 to NADP. Okay? So in here, this image shows you the overview of the flow of electrons during the light-dependent reactions of photosynthesis. So photosystem 2 actually boosts electrons from an energy level below that of water to a midway point. Okay? And then photosystem 1 raises electrons from a midway point to the energy level above that of NADP. Okay, so as you see, this is actually the energy content of the electrons, and then these are, the parallel to it, or um, in line with this content of electrons, energy content of electrons is actually the different molecules that are included in the different photosystems. Okay. So when the sunlight strikes a thylakoid membrane, energy is absorbed by the antenna pigments of both PS2 and PS1 and pass to the reaction centers of both photosystems. Okay? So electrons of both reaction center pigments are boosted to an outer orbital, and each electron is transferred to a primary electron acceptor. After losing their electrons, the reaction center chlorophylls of PS2 and PS1 become positively charged pigments referred to as the P680 and the P700 respectively. So the electron acceptors in turn become negatively charged after it. In essence, this separation of charge within the photosystems is the light reaction, which is the conversion of light energy into chemical energy. Okay, so again, there are always introduction of light and then targeting the reaction centers. Of course, there's always a, the transfer of these electrons. Okay, so this is how photosystem 2 and photosystem 1 actually work together. Okay. So, PS2 uses absorbed light energy for two interrelated activities. So, first, by removing electrons from water, and secondly, by generating a proton, or for the hydrogen gradient across the thylakoid membrane. So, PS2 of plant cells is complex of more, or it comprises of more than 20 different polypeptides, most of which are embedded in the thylakoid membrane. And two of these proteins known are actually the D1 and D2. So this D1 and D2 are very important because together they bind with the P60 reaction center chlorophyll dimer and all of the cofactors involved in the electron transport through the photosystem. Okay, so again, the two very important type of polypeptides are the D1 and the D2. Okay, so the first step in the PS2 or the photosystem 2 activation is the absorption of light by an antenna pigment. So light harvesting complex, or LHCII, is a pigment protein complex where most of the antenna pigment that collects solar energy for PS2 actually resides. Okay, so LHCII proteins bind both chlorophylls and carotenoids and are situated outside the core of the photosystem. So for you to be able to, go along with how the flow of electrons from photosystem 2 to plasstokinin happens, please check on the image that you will see on your book on page 208. So it would show you the functional organization of the photosystem too. So for the flow of electrons from photosystem 2 to plastoquinone, the first that you would need to understand is that excitation energy is passed from LHCII outer antenna pigments to a small number of inner antenna chlorophylls situated in the core of the photosystem 2. And from there, photosystem 2 reaction center pigment within around 3 picoseconds, the excited reaction center pigment P680 responds by transferring a single photoexcited electron to closely associated chlorophyll-like phyophytin molecule. So the phyophytin molecule is actually the first electron acceptor in the whole transfer. Okay, so the first electron transfer generates a separation of charge in photosystem 2 between a positively charged donor, which is the P680, and the negatively charged acceptor phyophytin. So because they are oppositely charged, we should know that they exhibit an obvious reactivity with one another because one is positive and then one is negative. So step 2, okay, for the transfer from photosystem 2 to phytoquinone, the phyophytin transfers its electron to a molecule of plastoquinone. It's denoted as PQ sub A, bound near to the outer stromal side of the membrane. So take note that plastoquinone or PQ is a lipid-soluble molecule similar in the structure of your ubiquinone. Okay, and then for step 3, the electron plastoquinone is transferred to a second plastoquinone, which is denoted as PQ sub B, to produce a semi-reduced form of the molecule that remains firmly bound to the D1 protein of the action center. With each of these transfers, the electron moves closer to the stromal side of the membrane. And then step 4, the positively charged pigment P680 is reduced back to P680, which primes the reaction center for the absorption of another photon, which sends a second energized electron along the path from P680 to phyophytin to plastoquinone to the semi-reduced form of plastoquinone, forming another plastoquinone reduced form, which is denoted as PQ sub B raised to 2 negative. Okay, so in step 5, this reduced form combines with 2 protons to form plastoquinol and PQH2 from the stroma, causing a decrease in hydrogen concentration of the stroma, which contributes to the formation of a proton gradient. And then step 6, PQH2 dissociates from the D1 protein and is released into the lipid bilayer and is replaced by a fully oxidized plastoquinone molecule derived from a small pool of plastoquinone molecules in the bilayer. Okay, so this actually, or these steps in the photosystem 2 would be easier if you would actually follow the image that you will see on your book. Okay, so you could read and follow the steps one by one. Okay, so here, this is the organization of the photosystem too. So these are the different parts. So I suggest that you read the steps and then trace it with the image that you see on your book or from this lecture for better understanding. Okay, so next, we have another one, the flow of electrons from water to photosystem 2. So the flow of electrons from water to photosystem 2 is actually the least understood part of the electron flow, where electrons come from water and are released when water is split into protons and molecular oxygen. So the splitting of water during photosynthesis is actually what you call your photolysis. Okay? So water is very stable. It's a very stable molecule made up of tightly held hydrogen and oxygen atoms. And splitting of water is the most thermodynamically challenging reaction known to occur in living organisms. So this type of thermodynamic connections are more likely endergonic. And the P60 or the oxidized P60 is most powerful oxidizing agent yet discovered in a living system. Okay? So, yet one photosystem 2 reaction center can generate only one positive charge P680 or oxidizing equivalent at a time. This problem has been actually solved by mechanism proposed by Pierre Hoyot and Bessel Koch around 1970. Okay? So, protons released during photolysis reaction are retained in the thylakoid lumen, adding to proton gradient, and the activity and integrity of the photosystem too can be negatively affected by high light intensity, a phenomenon termed photoinhibition. So there are too much light, or the too much light can diminish photosynthetic output by just like too much oxygen can. Okay? So, with the proposal of Pierre Hoyot and Bessel Koch, so they actually introduced what you call the S-state hypothesis, wherein it allows the photosystem to accumulate the 4 oxidizing equivalents needed to oxidize your water. Okay? So, to know more about this hypothesis or this proposal, you may actually read a part of it from your book. Okay. So next, we have the other process, or the other leg would be coming from the photosystem 2 to photosystem 1. So here we have the PQH2, which is a mobile electron carrier like your mitochondrial coenzyme Q. It can actually diffuse through the thylakoid membrane lipid bilayer and bind to a large multi-protein complex, the cytochrome B sub 6F. So, the successive absorption of 2 photons by photosystem 2 reaction center leads to the formation of the fully reduced PQH2, thus the production of one oxygen molecule. So, it requires the photosystem 2 absorption of 4 photons, leading to the formation of 2 PQH2s. Okay? So, cytochrome B6F is related in structure and function to cytochrome BC1 of the mitochondria electron transport chain. So, cytochrome B6F electrons pass to another mobile electron carrier, which is a water-soluble copper-containing peripheral membrane protein called as your plastocyanin, found on luminal side of your thylakoid membrane. So, all electron transfers in the process are actually exergonic, and electrons are passed to carriers with increasing affinity for them, so more positive redox potentials. For the summary of the steps in this stage of process, so again, we have PQH2 passes electrons to multiprotein complex of the cytochrome B6F, and then the hydrogen goes to the thylakoid lumen. Okay, so this is the first stage, and then cytochrome B6F passes the electrons to the plastocyanin on the thylakoid membrane luminal side. Okay, and then the plastocyanin then passes the electrons to the P700 oxidized P700 reaction center in the luminal side of the photosystem 1. Okay, so this is how the electron transport between photosystem 2 and photosystem 1 happens. So next, let's go now to the photosystem 1 operations. So it comprises with the process of the reduction of NADP and the production of NADPH. Okay, so photosystem 1 of higher plants consists of reaction center core made up of 12 to 14 polypeptide subunits and a peripheral complex of protein-bound pigments called LHCI, okay, or 1. So light energy is absorbed by the antenna pigments of the LHC1 or the light harvesting complex 1 and passed to the photosystem 1 reaction center pigment P700, which is a chlorophyll A dimer. So here you will see the schematic diagram of the functional organization of the photosystem 1. Okay? So after the light is absorbed by the LHC1, then an excited reaction center pigment P700 transfers an electron to separate monomeric chlorophyll A molecule, which acts as a primary electron acceptor. So you will be denoting it as A sub 0 or sub O. So the initial charge separation in the photosystem 1 is stabilized by moving electron from A sub 0 through several cofactors starting with a type of quinone called as phyloquinone. So that is denoted as A sub 1, and then 3 iron sulfur clusters known as F sub X, F sub B, and F sub A. So the electron is then transferred out of the photosystem 1 to a small water-soluble iron sulfur protein you call the ferredoxin, which is associated with the stromal surface of the membrane. Then NADP is reduced to make NADPH. So not all electrons passed to the ferredoxin inevitably end up in NADPH, and apparently there are other alternate routes that can be taken depending on a particular organism and conditions at a time. Okay? So again, for you to be able to follow the instructions of this photosystem 1 operation, I would encourage that you go back to the previous image that has been previewed and try to tackle each of the steps one by one. Okay, so for the overall summary, electrons go from water to NADP by the action of two light-absorbing photosystems while making oxygen and NADPH. So events in the photosystem 2 generate a strong oxidizing agent capable of producing oxygen from water, while events in the photosystem 1 generate strong reducing agent capable of producing NADPH from NADP. So at the same time, reduction of one NADP requires the transfer of two electrons, and photosynthetic light reactions also establish proton gradient across thylakoid membrane, which is the hydrogen removal from stroma in addition to the thylakoid lumen. And then it leads to ATP formation and these are actually the three contributors of the gradient. So for the summary of the light-dependent reactions, you could actually find it in your book on page 212. Okay? Okay, so let's move on now to photophosphorylation. So the light-dependent reactions discussed in the previous sections provide that energy required to reduce carbon dioxide to carbohydrate. Okay, so the conversion of 1 mole of carbon dioxide to 1 mole of carbohydrate requires the input of 3 moles of ATP and 2 moles of NADPH. So we have seen how plant cells produce the NADPH required for manufacture of carbohydrate. So let us now examine how these same cells produce the required ATP. So in photophosphorylation, it is the conversion of ADP to ATP using the energy of sunlight by activation of the photosystem 2. Okay, so again, it is the conversion of ADP or the adenosine diphosphate to adenosine triphosphate. So ATP synthesis machinery in chloroplast is virtually identical to that of a mitochondrion or aerobic bacterial plasma membranes. So as in those cases, the ATP synthase consists of a head called the CF1 in chloroplast, which contains the catalytic site of the enzyme, and AC base, or the CF sub 0, which contains or which spans the membrane and mediates proton movement. And the two parts are actually connected by a rotary stalk. So the various parts of the ATP synthase are made of or polypeptides that are homologous to those of the bacterial and mitochondrial enzymes. And the ATP synthases are not uniformly distributed throughout the thylakoid membrane, but are preferentially localized in stroma thylakoids and in exposed regions of grana stacks. So CF1 heads project out in the stroma, keeping with the orientation of the proton gradient, which has its higher concentration within the thylakoid lumen. So ATP produced in chloroplast by essentially same mechanisms used in the mitochondria. So here you have the proton gradient produced by pumping protons in the thylakoid lumen, and then protons go back to the stroma through ATP synthesis to make ATP. So it starts with the CF sub-zero, then through the head. And then isolated, illuminated chloroplast make medium alkaline as protons move into lumen. So let's tackle now the difference between the non-cyclic and the cyclic photophosphorylation. So for the formation of ATP during oxygenic photosynthesis, or what you also call your non-cyclic phosphorylation, electrons pass from water to NADP in a linear or non-cyclic path. So Daniel Arnon from the University of California in Berkeley in the 1950s discovered that the isolated chloroplasts were not only able to synthesize ATP from ADP, but could also do so even in the absence of added carbon dioxide or NADP. Okay? So this cyclic process, or the cyclic process for phosphorylation, is carried out independent of the photosystem 2 and begins with the absorption of a quantum of light by the photosystem 1 and transfer of a high energy electron to the primary acceptor. So the process that runs depends on the cellular needs of the ATP, NADPH, and your carbohydrates. So here shows you, or the image shows you here, the difference of the cyclic phosphorylation versus the non-cyclic phosphorylation. So again, with the non-cyclic, it's more likely just on a linear pathway, while with your cyclic, so, it goes back. Okay, the electrons, or the electron carriers are, moving in a circular pathway. Okay. So next, let's tackle now the carbon dioxide fixation and the formation of carbohydrates. So after World War II, Melvin Calvin of the University of California, Berkeley, along with his colleagues Andrew Benson and James Basham, began what was to be a decade-long study of the enzymatic reactions by which carbon dioxide was assimilated into the organic molecules of the cell. So, armed with newly available long-lived radioactive isotopes of carbon, or 14C, and a new technique they call the two-dimensional paper chromatography, they began the task by identifying all the labeled molecules produced when cells took up um, the radio-labeled isotope 14CO2. Okay, so in here, the study began with plant leaves but soon shifted to a simpler system, which is actually the green alga chlorella. Okay, so they used again, or they used uh, a simpler type of model organism. So they grew the algal cultures in sealed containers with cold carbon dioxide, then hot carbon dioxide for their desired incubation period. So the algae were actually a simpler system to work with than the plant leaves. So algae then drained into hot alcohol to kill algae immediately, stop enzymes and extract soluble molecules. So after which, they did a 2D paper chromatography on extracts and detected hot carbohydrates by autoradiography. So the C3 pathway was discovered when their findings with a labeled carbon dioxide plate was converted to reduced organic compounds very rapidly. So in a very short incubation period, about a few seconds, one radioactive spot on the chromatogram predominated, with which the 3-phosphoglycerate, or the PGA, one of the intermediates of glycolysis. So actually they found this PGA on one of their plates. Okay, so Calvin's group initially suspected that carbon dioxide was being covalently linked or fixed into 2-carbon compound to form the 3-carbon PGA molecule. So, however, because the first intermediate to be identified was a 3-carbon molecule, plants that utilize this pathway to fix atmospheric carbon dioxide are referred to as C3 plants. So, after further investigation, it became apparent that the initial acceptor was not a 2-carbon compound, but a 5-carbon compound, which is what they call the ribulose 1,5-bisphosphate, or the RUBP. Okay? So this is actually condensed with with carbon dioxide to produce a 6-carbon molecule, or 2 molecules of PGA, where one contained a recently added carbon atom. So both the condensation of carbon dioxide to RUBP and the splitting of the 6-carbon product are carried out with the stroma by a large multi-subunit enzyme, ribulose bisphosphate carboxylase oxygenate, or what you call, or oxygenase, or what is commonly known as the Rubisco. Okay? So coming from the initial findings of a 2 or a 3 carbon molecule compound, they eventually, found out that it actually changed, okay, from a 3 to a 5 molecule, which is the Rubisco. Okay. So as the structure of the various intermediates was determined, together with the positions of the labeled carbon atoms, it became apparent that the pathway for conversion of carbon dioxide into carbohydrate was cyclic and complex, that occurs in cyanobacteria and all eukaryotic photosynthetic cells. And this pathway is known as the Calvin cycle, or the Calvin-Benson cycle. So the series of reactions forms a Calvin cycle is composed of 3 main parts. The first one would be the carboxylation of your RUBP to form PGA. And then the second one would be PGA reduction to the level of sugar to form glyceraldehyde 3-phosphate, or the GAP, using ATP and NADPH made by light-dependent reactions. And lastly is the regeneration of RUBP, which also requires additional use of ATP. So this image here on your screen right now is actually showing you how the conversion of carbon dioxide into carbohydrate happens in the form, or in the process of the Calvin cycle. Okay, so with this one, please go through with the steps one by one and trace, at least even create your own, diagram, wherein you'll be able to easily understand the pathway. Okay, so balancing the cycle, what happens when 6 molecules of carbon dioxide are fixed by Rubisco? So it can be seen in figure 16.9 that has been presented earlier, that for every 6 molecules of carbon dioxide fixed, 12 molecules of GAP, or glyceraldehyde 3-phosphate, are produced, and atoms in 10 of them are arranged to make 6 molecules of the 5 uh, carbon dioxide acceptor, RUBP. Okay, so the other 2 gaps are products which can be exported to the cytosol in exchange for phosphate ions, where they are converted to be disaccharide sucrose. And alternatively, GAP can remain in the chloroplast where it is converted to starch. So an overview of the entire process of the photosynthesis, including the light reactions, phosphorylation of ADP, and the Calvin cycle, and the synthesis of starch or sucrose, is shown in figure 6.20. Okay. So sucrose molecules that are formed in the cytosol from the gaps of the Calvin cycle are transported out of the leaf cells and into the phloem, where they are carried by various non-photosynthetic organs of the plant. And then the conversion of the 6 molecules of carbohydrate or carbon dioxide to one 6-carbon sugar molecule, and the regeneration of RUBP requires 12 molecules of NADPH and 18 molecules of ATP. So this large energy expenditure reflects the fact that, carbon dioxide is the most highly oxidized form in which carbon can occur. So next we have the redox control. So with redox control, this is actually a regulator of the basic cellular processes including protein folding, transcription, and translation. So, and it is also best understood as a regulator of the chloroplast metabolism. So, several key enzymes of the Calvin cycle are only active in the light when ATP and NADPH are being produced by photosynthesis. So, this light-dependent control of chloroplast enzymes is exercised by a small protein called the thioredoxin that can occur in either a reduced or oxidized form. So it is noted that not all of the electrons that pass through ferredoxins are used to reduce NADP because some of them are transferred to thioredoxin. So once it has accepted a pair of electrons, thioredoxin reduces certain disulfide bridges into sulfhydryl groups in selected Calvin cycle enzymes. And this covalent change in this protein structure activates these enzymes, promoting the synthesis of carbohydrates in the chloroplast. However, in the dark, when photosynthesis has ceased, thioredoxin is no longer reduced by ferredoxin, and the Calvin cycle enzymes revert to an oxidized disulfide bridge state in which they are inactive. So here, you will see how the redox control of the Calvin cycle happens. Okay, so with the presence of the light, how does your ferredoxin actually is being reduced? So for the next process, we have what you call your photorespiration. So photorespiration is actually, a wasteful pathway that competes with the Calvin cycle. So it begins when the rubisco acts as an oxygen instead of the carbon dioxide. So while glycolate is not part of the Calvin cycle, it is a product of a reaction catalyzed by rubisco. So approximately 20 years after it was found that rubisco catalyzed the attachment of um, carbon dioxide to RUBP, it was discovered that rubisco also catalyzed a second reaction in which oxygen is attached to RUBP to produce 2-phosphoglycolate along with PGA. So phosphoglycolate is subsequently converted to glycolate by an enzyme in the stroma, and this glycolate is produced in the chloroplast and is transferred to the peroxisome and eventually leads to the release of carbon dioxide. Okay, so basically with this one, there is a higher chance of you having, or a higher chance of having carbon fixation. So because respiration involves the uptake of oxygen and the release of carbon dioxide, this series of reaction is actually what you call your photorespiration. Because photorespiration leads to the release of previously fixed carbon dioxide molecules, it is a waste of the plant's energy. So photorespiration can account for the loss of up to 50% of newly fixed carbon dioxide by crop plants growing under hot, dry conditions. So In here, C4 and CAM plants are found in hot, dry environments where conditions favor the wasteful photorespiration reactions of rubisco, or when either water or carbon dioxide is not or is in short supply. Okay, so basically these are um, more useful when it comes to those plants that could actually thrive or grow wherein there's so much or there is a height in temperature, and of course there is a limitation when it comes to the availability of water. Okay, so the C4 and CAM plants will be discussed further as we go along with this lecture. So next, for peroxisomes and photorespiration, so photorespiration begins when RUBP reacts with oxygen instead of carbon dioxide to form 3-PGA and 2-carbon phosphoglycolate. So once formed, phosphoglycolate from photorespiration is converted to glycolate, which is shuttled out of the chloroplast into a peroxisome. In the mitochondrion, two glycines, which is actually a 4-carbon molecule, are converted to one serine, a 3-carbon molecule, and they release one molecule of carbon dioxide. So, two groups of plants, C4 and CAM plants, have overcome photorespiration's negative effects by evolving metabolic mechanisms that increase the carbon dioxide and oxygen ratio to which rubisco molecules are exposed. So, this is how, different plants could actually evolve because of, the different factors, no, that could affect their growth and development. So this is a representation of how photorespiration and photosynthesis actually look like. Okay, so as we know, photosynthesis is actually using carbon dioxide to be able to produce or to have or to produce its food or for the food of the plants. In photorespiration, it actually uses the oxygen. Okay, and it actually produces more carbon dioxide. Okay, so basically this photorespiration is more likely to use more ATPs, hence it's thought to be more wasteful compared to your photosynthesis. So in photosynthesis, you will be generating you know you will just be using one ATP at a time to produce food, but in photorespiration there could be multiple times wherein ATP is used, and of course carbon dioxide is not also very favorable to other organisms, no. So basically, yeah, photorespiration is a type um of a process wherein it's a wasteful type of using available energy sources. Okay, so the carbohydrate synthesis for C4 plants. So in 1965, Hugo Korczak reported that when sugarcane was supplied with the hot CO2 radioactive isotope, its radioactivity first appeared in organic compounds containing 4-carbon skeletons rather than the 3-carbon PGA molecule found in other types of plants. So, hence, further analysis revealed that these 4-carbon compounds, predominantly the oxaloacetate and malate, resulted from the combination of carbon dioxide with phosphoenol pyruvate or PEP, and thus represented a second mechanism of fixation of atmospheric carbon dioxide. Okay, so structure and the function of the C4 plants could be viewed or could be reviewed from your book on page 218. So, the enzyme responsible for the linkage of carbon dioxide to PEP was named phosphoenol pyruvate carboxylase, and it catalyzes the first step of the C4 pathway. So, plants that utilize this pathway are referred to as C4 plants and are represented primarily by tropical grasses. So this example, or the examples that you could actually think about the C4 plants are those that are being planted during or in the farms, okay, like maize, okay, you have sugar cane, so those are types of C4 plants. So why did the C4 photosynthesis evolve? So if a C3 plant is placed in a closed chamber and its photosynthetic activity is being monitored, it is found that once the plant reduces carbon dioxide levels in the chamber and the rate of carbon dioxide released by photorespiration levels at the rate of carbon dioxide by photosynthesis, the net production of the carbohydrate stops. Okay? So basically there is, a monitoring here of how the carbon levels or the carbon dioxide levels should be monitored. Okay, so if they drop too low, this or the production of these carbohydrates could actually stop. Okay, so C4 plants have this ability to continue photosynthesizing because of PEP carboxylase that continues to operate at much lower carbon dioxide levels than does rubisco and is not inhibited by oxygen. So carbohydrates production continues until your carbon dioxide drops up to 2 or 1 to 2 parts per million. So the C4 pathway value is apparent when C4 plants are placed in dry, hot environment like that in which of many of them could actually live. So even if there's, a difference with the amount of carbon dioxide because of the PEP, they can still do the photosynthetic thing until, carbon dioxide is no longer available. So in hot, dry climates, C4 plants have big advantage and C3 plants lose much of water through transpiration. So in such a climate, plants lose water through openings you call the stomata at the surfaces of their leaves. C4 plants can keep their stomata closed to prevent water loss, but they can still maintain sufficient carbon dioxide uptake to fuel photosynthetic activity at a maximal rate. The carbon dioxide fixed to PEP is soon released and captured by rubisco, after which it enters the C3 pathways and is converted to C3 metabolic intermediates. So, a number of plant science labs are trying to introduce parts of the C4 photosynthetic machinery into C plants in an attempt to boost crop productivity. So the PEP carboxylase gene from maize AC4 plant has been introduced into rice AC3 plant in hopes of recreating or creating a carbon dioxide concentrating mechanism within rice mesophyll cells where rubisco is housed. However, it is still not known if observed increases of photosynthetic efficiency are due to higher levels of carbon dioxide assimilation or lower photo respiration levels, higher stress resistance, or some other mechanisms. So, the studies of this uh, type of introducing genes to other plants are actually or would still take long, no, for them to be able to produce such kinds of rice that could actually do or that could actually um, maintain their growth even if there is lack of water and of course and the high temperature. Okay, so C4 plants are thought to have evolved fairly recently, about 7 to 10 million years ago, when carbon dioxide levels were very low. So next, and the last for our discussion about photosynthesis are about the carbohydrate synthesis in plants. So many desert plants such as cacti or the cactus have another biochemical adaptation that allows them to survive in very hot and dry habitats. So these plants are what you call your CAM plants, and they utilize the PEP carboxylase in fixing atmospheric carbon dioxide just like your CIFAR plants. So for CAM species, they carry out light-dependent reactions and carbon dioxide fixation at different times of the day rather than in different cells of the leaf. In other words, evolution has followed two different pathways in overcoming the problem that photorespiration possesses as it decreases photosynthetic efficiency. So, CAM plants keep their stomata tightly closed during the hot and dry day and open at night when they fix carbon dioxide with PEP carboxylase and when the rate of the water vapor loss is greatly reduced. So, during nighttime fixation of carbon dioxide in mesophyll cells, malate is made and transported across the tonoplast membrane into cell's central vacuole. Malate's presence is evident by sour morning taste of plants. So during the day with the stomata closed, the malic acid is moved into the cytoplasm. Okay? So for the references, you may actually read again the second part of the cellular respiration and photosynthesis from your book, which is by Carp. If you have any questions for me regarding this lecture, please let me know and I'll be answering them. Thank you very much and have a great day.